Hello, welcome to the presentation of our work, Multiple Layer, Multiple Patterning Aware Placement Refinement for Mixed Cell Height Designs. This work is done by Chi-Chun Fang, Professor Ting-Chi Wang, Professor Wai-Kei, and me. There are three main placement issues that we need to deal with: MPL, interval conflict, and conflict interplay among layers. The first placement issue is multiple patterning lithography. With the use of MPL, the circuit patterns are partitioned into several different masks to get lower pattern density in each mask. Layout decomposition is formulated as a graph coloring problem. After the coloring method is applied to the conflict graph, features are assigned to different masks according to the coloring result. The second issue is the interval conflict. If the features on the layer are separated by the PG rails, there is no interval conflict. On the other hand, the features that are not separated by the PG rails may cause an interval conflict. The third issue is conflict interplay among layers. In a multi-layer MPL problem, the refinement method that solves the conflict for a layer may lead to the occurrence of a new conflict in another layer. Therefore, how to solve the conflict without creating the new ones is important. Previous work has tackled the placement and multiple layer MPL layout decomposition problem. It targets a special case considering TPL for the M1 layer and the MOL layer. In this paper, we address a general problem of simultaneous mixed cell height placement refinement and multiple layer MPL layout decomposition, where interval conflicts can occur in an arbitrary manner. Here's the problem we try to solve. Given a legal placement with single row height cells or multiple row height cells, and a minimum coloring spacing rule for each MPL layers, where different layers have different requirements on the number of masks, the problem asks to find the legal placement within given placement region, as well as the layout decomposition for each MPL layer, such that the number of the coloring conflicts and the total cell displacement are both as small as possible. UCG identification is the method that we use to find the cells that we need to refine. If a kinetic component is not K-colorable, our coloring method will not assign a color to any of its vertices. Each uncolorable kinetic component indicates a group of uncolored features, and the set of cells that contains these uncolorable features is called uncolored cell group. In this picture, our coloring method will not color for the 4 features circled. Instead, our method takes all features that lead to the coloring conflict into consideration, and the corresponding cells A, B, and C form a UCG. Our methodology contains 2 main parts: initial coloring and MPL aware placement refinement. In the initial coloring phase, a conflict graph is first constructed for each MPL layers to transform those features into a graph representation according to the given minimum coloring spacing rule. If there are uncolorable features in the conflict graph, our method will identify a set of uncolored cell groups which contain cells with uncolored features. The MPL-aware placement refinement phase will try to eliminate as many UCGs as possible. If there are still unsolved UCGs, the UCG simplification is used. The UCG simplification stage tries to divide the remaining UCG into the small ones. This table shows the refined result by previous work and by our methodology. As can be seen from the table, previous work alone cannot produce a placement without any coloring conflict for all test cases but one. Therefore, our methodology is a necessary complement to previous work, as it can eliminate all remaining coloring conflicts for all those test cases. In this paper, we present a mixed cell height placement refinement methodology for multiple layer MPL. The concept of UCGs is introduced and employed by our methodology to help identify cells which cause the coloring conflict. The experimental results shows that our methodology can effectively eliminate UCGs and hence the coloring conflicts with a very small cell displacement. And that's all for my presentation. Thanks for listening.